Hey viewers, welcome to my practical bioinformatics series where I talk about tools and techniques of bioinformatics that I employed. In this video, I will talk about antigen-antibody docking that I did as part of a lab rotation at Stony Brook University in the time frame of February 2019 to May 2019. Hello, I'm Arpita Banerjee and welcome back to my channel. In this particular video, I'm going to walk you through another of my rotation projects. I could spend the least time in the lab for this particular rotation. The reason being, I was attending a lot of classes and also I had teaching responsibilities. But despite that, I think I found something substantial and I'm going to share my findings with you. Something I should clearly mention is that all the rotation projects which I'm presenting, I did not choose the projects per se. They were handed over to me. I chose the course of my work. This video is on antigen-antibody docking, where I will discuss a simple visualization technique that tells volumes if you know what you're looking for. So let's begin. Now, what is an antigen and what is an antibody? Both are proteins. Antigen often comes from an outside source, such as pathogen. Antigens are actually present on blood cells as well, but we will keep this video simple. And so simplistically, antigen comes from a foreign source, such as pathogen. An antibody is a protein that comes from the host's immune system, and the host could be you, a human. Antibodies react or interact with antigens, and in this process, they essentially block the antigen and gives rise to further downstream immune response per requirement. An example of an antigen could be the envelope or the code protein of a virus, and the antigen antibody interaction that you see over here takes place along the CDR or complementarity determining region on the antibody, the shaded green part that you see over here. So, not all of the antibody and not all of the antigen are involved in the interaction. Sometimes the immune system of the host can detect the antigen right away, that is, if the host has been pre exposed to the virus. If not, antibodies are produced in response to a novel virus attack. The interaction takes place along CDR in green on the antibody and on a tiny region of the antigen shown in red here. Now, if you consider this to be the case with strain 1 of the pathogen, in case of strain 2, the tiny portion of the interacting antigen undergoes change. Notice the color change from red to blue. The hypervariable region or the CDR of the antibody undergoes change in response. Notice the change in color to red when it comes to strain 2. When you are attacked by another pathogen altogether, then the interaction varies a lot. That is to say that the epitope and antigen and the CDR of antibody differs much more as compared to the case of two different strains from the same pathogen. Now, there's something to know about this antigen antibody interaction. There are certain amino acids that actually make up the epitopes, and that also applies to the antibodies. There are certain amino acids that make up the hypervariable region. And of course, with different pathogens or different strains of the same pathogen, there are variations within that zone. There have been books written on the sequences, software built on these sequences. We will not cover those in details in this particular video. So I gave you a broad overview of antigen-antibody interaction and how the immune system deals with pathogen attack. Now, why bother about all that I told you? Because they have applications in vaccine development. What happens in case of a vaccine is that a dead or attenuated pathogen, that is a dead or a weakened pathogen, is introduced into the host. The host's immune system gets to know the pathogen, it gets to know the antigens from the pathogen, and it produces antibodies in response to that. Right? So the immune system gets prepared, whereas the pathogen being dead or weakened is unable to cause much harm to the host. So what happens is when the live pathogen actually attacks the host, the immune system is already prepared and the response of the immune system is much faster. So that way, the damage to the host is much less or null. So my work entailed antigen-antibody docking, where I got the three-dimensional coordinates of the antigen from a crystal structure deposited in PDB. And for the antibody, I got its 3D coordinates by building a homology model based on the sequence of the antibody that was in the same PDB structure I mentioned. Finally, I docked the two, the antigen and the antibody. You could have taken crystal coordinates for both of them without building a homology model for the nanobody. That is always an option. My computational work involved homology modeling and the docking. I knew, however, that I was trying to simulate antigen antibody interaction, which means that the docking had to be along the lines of the CDR and the epitope. Okay, so I worked with this system that you see over here. It is not a symbolic representation, but rather this is a representation from the crystal structure 4AQ1 deposited in PDB. The antigen is demarcated on top. It is the SBSPS layer protein of Geobacillus styrothermophilus, and nanobody KB6 is right below. Something to notice in the 4AQ1 structure is that the antigen in green here is quite large and it looks almost like antibody, whereas the nanobody here is in orange. And if you see the description here, you will see that SPSB from the pathogen consists of a 7-domain protein formed by an amino terminal cell wall attachment domain and 6 consecutive immunoglobulin-like domains. So that's the reason why the antigen in green looks like antibody because of the immunoglobulin-like domains. And if you see further below, SPSB protein has this image, which is almost like antibody, whereas the nanobody NBKB6 is quite smaller in comparison. Nanobodies are actually target binding fragments of monoclonal antibodies. So they essentially are like antibodies, but they have enhanced chemical properties or enhanced selectivity. So they could be used for protein purification as well. Maybe this nanobody over here was used to precipitate down the SBSB layer of this particular pathogen, which aided in crystallization of the protein. In this video, we will refer to the nanobody as antibody to make it sound simpler. I specifically worked with the shaded regions that you see over here. This is because, as you can see, and as I had mentioned earlier, not all of the antigen and antibody are involved in the actual interaction. Right. So if you notice, it's just a small part which is interacting with each other. So the rest of the structure is not relevant. So I did not include them. And I was actually made to model the pink shaded region, that is the antibody. Before I submitted my docking jobs, it was just for exercise purposes. I needed the antigen structure and the antibody structure, right? So for the antigen, I took the coordinates from the 4AQ1 crystal that you see over here shaded in blue. And for the antibody, I got its 3D coordinates by building a homology model using the protein sequence from part of the structure, which is shaded in pink here. Now, while searching for templates for homology modeling, these are the hits that came up, the PDB IDs of which are on the extreme left. By the way, templates are structure from which the 3D coordinates are copied. Sequence identity is very high in all the cases, as you can see. The table shown here has certain other parameters as well, which could help you decide on the template. But with this high level of sequence identity, you could expect good models for all of them. The minimum sequence identity for homology modeling is around 30%. And here we have like 82 point something for all of them. So I went ahead and selected the first hit 5IMK for building my homology model. I used a web server of Swiss model to build my homology model. Now, upon comparing the structure of the model with respect to that of the template, this is what I got. The template is in green here and the model is in light blue. It's almost complete overlap with RMSD between pruned atom pairs 0.113 angstrom. RMSD is root mean square deviation. It is a quantitative measure of telling you how much deviation is there between the structures. And as you can see, and as the RMSD value tells you, it is almost a very close match. One can opt to take the antibody coordinates directly from the structure as well. The structure in this case is 4AQ1, instead of modeling the antibody. So how much does the homology model, differ from the crystal coordinates of the antibody, which is shaded in pink here? This much. The RMSD between the pruned pairs is 0.931 Armstrong. The model is in light blue and the crystal structure is in light brown here. So as you can see, it is not that one is an apple and the other is an orange. So if you dock the homology model with the antigen crystal coordinates, it could be expected that the interaction between the antigen and the modeled antibody would be somewhat like this, as has been freezed in the crystal structure. Now, going to the ClusPro web server for docking job submission, receptor and ligand had to be specified. For the receptor, the PDB file of the built homology model was uploaded, and for the ligand, the PDB coordinates of part of the antigen was uploaded. And I'm talking about this part that you see shaded in blue here. The coordinates of only this part was taken from the crystal structure. And antibody mode was selected for the docking with the option automatically mask non-CDR regions. Now, what are these non-CDR regions and why were they masked? Going back to the typical symbolic representation of antigen-antibody interaction, look at the antigen binding fragment of the antibody. This is the CDR or complementarity determining region. And this is the only region that interacts or makes contact with the antigen. The rest of the structure does not. And therefore, the rest of the structure or non-CDR regions were masked. Only the CDR region was made available for docking. Okay, now how to check the docking results? There must be some reference to compare your results to. So to check my docking results, this is what I did. I colored part of the antigen in yellow and the antibody in red in the 4AQ1 crystal structure. Notice I colored only those regions that are interacting with each other because that is of relevance to us. You know, the antigenic phase and the CDR of antibody coming together. This is my reference. The amino acid residues I colored are clearly mentioned. So I was checking my docking results to see if these are the residue stretches that are coming close to each other in the same orientation as you see in the crystal structure. ClusPro returned 30 docked confirmations. And this is the first one of the lot. Now, as I will move on, the yellow will be pointed out with this blue arrow and the red will be pointed out with this red arrow. I have taken the docking confirmation picture slides from the presentation report that I had sent to the lab PI back in 2019. Keep an eye on the arrows. So this is the first one of the lot. And then you have this, next, 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 next. So up till now, notice that the binding regions, the colored regions or the colored arrows, they are not coming together as they were supposed to. Next, 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 again. None of the docked confirmations showed interactions similar to that in the crystal structure, considering the reference points. So before even I actually started working on the project, the PI of the lab had mentioned CASP. CASP is Critical Assessment of Structure Prediction. It's a competition where computational biologists, they submit their predicted structures. The actual solved structure is held back. It is not known to the ones who are predicting the structures. The structure could be a homology model, could be docked complexes. So the actual structures are released only after all the predictions get submitted. With the results I showed you, it goes without saying that I would not have won the competition. Now coming back to the reference structure, look at the colored reference points again. These are the regions that should have docked, but they did not. I'm not claiming that the yellow is the epitopic region on the antigen and the red is the CDR region on the antibody, but yes, they're very likely so. And determining CDR via masking the non-CDR region was the job of the software. That probably didn't go well. If you notice the dot conformations, the interaction region between the antigen and the antibody, it is all over the place, all over. I mean, just take a look at the orientations and you will know what I am trying to say. So the interaction region is all over the place. The docking was not taking place along the CDR. Definitely that was not happening. And when I showed these results to the lab PI, it appeared to me that he was not expecting this. I don't know how they checked their results for this particular system and if they checked it right at all. He mentioned biological assembly to me. Now, what is biological assembly? As per PDB's site, biological assembly is the macromolecular assembly that has either been shown to be or believed to be the functional form of the molecule. In a crystal structure, biological assemblies are derived by symmetry operations consisting of rotations, translations, or their combinations. You can see the biological assembly of hemoglobin shown here for three different PDB structures. If you notice, in case of hemoglobin, regardless of biological assembly, your interaction between the different chains is not going to change. I guess it is very clear from this picture itself. In the 4AQ1 entry of PDB, if you look on the left, you have biological assembly 1, and then 2, 3, 4. This is the symmetric unit. So if you click on this 1D 3D view, you will be able to see the structure in three-dimensional view in the PDB's site itself. So here it is. Now let me just show you something. If you hover your cursor along the structure, like if I hover along the residue stretches that are very near to the antigen, so you could say that, you know, they are forming part of the interface. I'm talking about these residue stretches. And notice that at the corner at the bottom right, you will see that the residue number and the exact residue is being mentioned. Say, for example, right now I'm at a spot which is tyrosine 59. Now, if I move along a little up, it is tyrosine 60. So this is the way you will actually be able to pinpoint the residues which are forming part of the interface. If you're unable to see the bottom right corner in this video, then I suggest that you go to PDB and view the biological assembly for this particular structure. You can download the biological assembly files from here. If you just scroll down, you will see that you can download the files from here. This is biological assembly 1 from the 4AQ1 record of PDB. Now, if you compare this structure with the structure that I worked on, you can clearly see a difference. And the minute details will be more clear to you if you notice the green balls over here. These are actually calcium ions. And notice that these two, which are like sort of close by here, so follow my cursor movement, these two calcium ions, they appear closer to the antibody structure over here in the biological assembly 1 compared to the crystal structure that I worked on. Here you will see that the antibody or the nanobody is down here below, whereas the two close by calcium ions are way up. So the orientation is very different. So we are not talking about the same interactions actually. That is to say, the intermolecular interaction between the antigen and the antibody are not same. They are different in the structures. Okay, here's an exercise for you. Observe the biological assembly structures. You could either do that in PDB or you could download the structure and view them in any visualizer. Note down the residues that form the interface because the interface is formed by epitope of the antigen and CDR of the antibody. Download the biological assembly structures, obviously one at a time. Mark the residues in the interface by coloring them differently. That would be your structural reference. So essentially, I'm asking you to do the same what I did for the main PDB structure they have for that entry 4AQ1. Mark the residues in the interface by coloring them differently because that would be your structural reference. Now separate the antigen and antibody structure and submit them for docking in the ClusPro web server with the specifications as mentioned earlier, like masking the non-CDR regions and stuff like that. Download the results having the different conformations and view them. Search the reference motifs in each docked conformation and color them as before. You have to observe whether the docked conformations are actually structurally like the way it is in the reference structure, the biological assembly structure. That's the exercise for you. Repeat it for all the biological assemblies. There are 4 of them. Now, before you delve into the exercise, I have some other suggestions for you. I'd say check out other PDB entries for the same biomolecule. Check out PDB for structures of nanobody with the SPSP layer of the same pathogen and see if the main PDB file structure differs from that of the biological assembly. And number 2, predict CDR epitope using some other software and take a consensus. This is to zoom in onto the exact CDR and the epitope region. The reasons for my mentioned suggestions are as follows. I have reservations about the difference in intermolecular interactions that is being observed between the main PDB file and the biological assembly structure files. So that is why I'm asking you to check other PDB structures. And number 2, ClusPro does not explicitly mention the regions that it considers as epitope and CDR. Okay, so that those calculations go on in the background, at least when you're using the web server. So make predictions using other software to confirm that the regions at the interface, as seen in the structure, are being predicted as epitope and CDR as well. This is again to counter the anomaly that is being observed between the main structure and the biological assembly. Okay, something I should mention very clearly is that when you're downloading a structure, it is the normal regular protocol to download the PDB format file that you see over here that actually I had downloaded and worked on, the results of which I showed you. Now for biological assembly 1, this is the structure where the antigen is in yellow and the antibody is in red. Now if I zoom in, the interaction along the interface is via these stretch of residues that make up this beta sheet over here, threonine 58 to tyrosine 60. This is for the antibody. Okay, for the antigen, the residues are lysine 490, arginine 491, and so on. For now, we will just talk about the antibody. Considering the intermolecular interactions in the biological assembly 1, we will try to see if the software had been able to detect the 58 to 60 residue stretch, the beta sheet stretch on the antibody as the CDR. Now, whatever I'm going to show you and tell you further down is based on these 2 assumptions. The biological assembly structures are correct. Now, they have to be correct because they will serve as my reference structure. They will point out where the CDR is and where the epitope is. Okay, so it is very important that the structures are correct. And number 2, CDR detection via masking of non-CDR regions precedes docking. And the process is independent of antigen structure that the antibody encounters while the docking happens. Now, the final docked conformations, they are subject to favorable interactions with the antigen. Okay, that is one thing. But what I'm trying to say, if you find the mentioned beta strand of the antibody oriented towards the antigen, it means that it has been considered as CDR and has been presented as CDR for docking. Now, coming back to my docked conformations, it is noticeable that I have shadowed out the antigen. The reason being, I'm just considering the antibody here because I worked with the main PDB structure. The part of antigen which is reacting with the antibody here in biological assembly 1 is not the same as in the monomer crystal structure, right? So I'm not considering the antigen. Okay, so just focusing on the antibody and seeing where the CDR is going, the little beta sheet structure that I showed you. Definitely, it is not being presented as CDR when it comes to this particular confirmation. No. In 4, yes, if you notice the black arrow, then yes, this part is being presented as CDR. In 5, a little bit of it, yes. In 17, a little bit of it, yes. In 19, again, a little bit of it, yes. 23, yes, this part is coming up as CDR visibly. In 25 too. In 27 as well. And in 28 as well. So whatever I told you with regard to the CDR on the biological assembly and based on my earlier docked confirmations, these have to be taken with a grain of salt for very obvious reasons that they have validity only if the assumptions I mentioned hold true. So what was the lesson learned from all this? Checking your output is necessary. That is to say, you cannot blindly trust the software. You cannot just rely on the computer and the simulations to do the job and just forget about it. No, you have to check your results manually to see what's going on. And if you spot errors like I did, you have two choices. Either you fix it or you change software. Wish you all a very happy new year. It's a coincidence that the day I'm recording this today, it is 1st of January 2023. Thank you.